So the organism that we use to study transcription regulation is the yeast Saccharomyces cerevisiae. Yeast is a single cellular organism that can grow on rich media. The yeast that we're using is actually called baker's yeast and is involved in making bread, and it's one of the common yeasts that making bread or making beer. We use yeast because it serves as an excellent model system for higher eukaryotes, including humans. Yeast is a good model system because it's cheap media or inexpensive media. It has well-developed genetics. We can knock out genes. We can introduce new genes back into it. The genome's been completely sequenced. It's relatively small. But most important is that many of the genes in yeast are highly conserved with human genes. And so our approach is that we try to understand how genes are regulated and how these proteins work in yeast, and that serves as a model for understanding how the proteins in humans may work as well. What our approach has been is that we use a combination of genetics, molecular biology, and biochemistry to understand how these proteins work that regulate gene expression. And so some of the tools that we can use is that we can go in and make mutations in a specific gene that codes for a protein, and then see how that mutation affects the protein's activity. And so we can see whether it knocks out the protein, or whether it no longer functions, or whether it now functions better. And so we can do that in the genes that code for the proteins, or in our case, our proteins are recognizing specific sequences of DNA to turn on and off genes, and so we can mutate the sites that they bind to as well. Transcription, one of the reasons why we study transcription is one of the fundamental processes in biology. For example, every cell within a human contains the same genetic information. But clearly those cells are different functions. The heart cell has different functions than a brain cell or a liver cell. And so why are there these differences? Well, clearly is that there are genes that are expressed in the heart that are not expressed in the liver or the brain. And so one of the things we're trying to understand is that what regulates those expression of genes within specific cells. Now, the way genes are expressed is that there is a polymerase, an enzyme called RNA polymerase, that binds to specific DNA sites upstream of the gene, and then they transcribe that DNA into RNA that then eventually gets translated into protein. Now, RNA polymerase, although it can carry out this process of transcription, it requires other factors to be recruited to the promoters or to the genes itself. So, for example, here, RNA polymerase won't bind to the site on its own, but if there's a factor that binds upstream to that site that will recognize the DNA site, it will bind and help recruit RNA polymerase, therefore turning this gene on. And so these activator proteins, these proteins that recognize specific sites upstream of the gene, are very important for regulating whether a gene is off or on. So in the absence of the protein, the gene is off. In the presence of the protein, the gene is on. And so these are called activator proteins. Just like there's activator proteins, there's also repressor proteins. And they work by also binding to sites. And they have the opposite effect in that they prevent polymerase from binding to the promoter and therefore turning off genes. So it's this interplay between activators and repressors within a given cell type that will determine whether a gene is on or off. And so what we're interested in is understanding how do these regulatory factors, these repressors, these activators work. How do they recognize specific sequences, and how do they prevent polymerase from working?
For more information on Andrew Verschan and his research, please visit waxman.rutgers.edu/wax/Verschan.